Welcome back to this next video, and in this video we are going to talk about the extrinsic pathway, which is also known as the death receptor pathway. Before going into the details of the extrinsic pathway, let us discuss briefly about the apoptosis. Now this apoptosis, it's from Greek meaning falling off, and is a process of the programmed cell death that occurs in multicellular organisms. During the process of the apoptosis, those cells in the body which have performed their functions and are no more able to perform normal physiological functions in the body, they are removed from the body. Therefore, we call them as the programmed cell death. During the process of the apoptosis, certain biochemical changes that occur, for example, one of the important biochemical change that is known as the blabbing, the cell shrinkage, the fragmentation of the nucleus, the condensation of the chromatin, the chromosomal DNA fragmentation, and global messenger RNA decay. Now, these biochemical changes that actually leads to the degradation of the cell and hence their removal from the body. The importance of the apoptosis for the human being is obvious from the fact that between 50 to 70 billion cells that die each day due to apoptosis in the average human adult body. So if you are removing 50 to 70 billion cells from the body each day by a process, that means that this process is one of the most important processes for the normal human being and for the normal physiological functions of the human body. Now the apoptosis that is very different from another process which is called is the necrosis. Now in necrosis, the cell damage occur and those cells that are removed from the body, but the process of the necrosis that occur because of the acute cellular injury. For example, if there is acute cellular injury to a cell which still have the ability to perform its function, but because of this acute cellular injury it is badly affected, and hence the cell that is removed from the body. While in the process of the apoptosis, a cell completes its normal life, and when it completes its normal life, then it is removed from the body by the process of the apoptosis. So the necrosis and the apoptosis, you can say that necrosis is an unwanted process, the apoptosis is a wanted process for the normal human beings. Now during the process of the apoptosis, specialized apoptotic bodies that are formed, and the advantage of these apoptotic bodies is that the phagocytic cells, they are able to engulf these apoptotic bodies and remove them because before the content of the cell can spill out onto the surrounding cells and cause damage to the neighboring cells. Because these apoptotic bodies that can have proteases that have the ability to degrade the surrounding cells, therefore these phagocytic bodies, the phagocytic cells, they engulf them, thereby preventing the surrounding cells from the proteases that can be present in these apoptotic bodies, and hence the surrounding cells that are not affected due to these apoptotic bodies. There are two pathways for the apoptosis that you can observe in the human beings. One of the pathway is known as the intrinsic pathway. Now in intrinsic pathway, the cell kills itself because it senses the cell stress, and the signal for the killing of the cell comes from the interior of the cell. If you talk about the extrinsic pathway, in the extrinsic pathway, as the name indicate, this is extrinsic, that means that the signal for killing the cell that comes from outside the cell and not from the interior of the cell. Therefore, this is the intrinsic pathway in which the signal that is generated within the cell for the death of the cell, and in the extrinsic pathway, the signal comes from the exterior of the cell, not from the interior of the cell. In both of these pathways, whether we talk about the intrinsic or the extrinsic pathway, in both of these pathways, the cell death is induced by a specialized class of the proteins, the enzymes which are known as the caspases, and these caspases are actually proteases which have the ability to degrade the proteins. Now, in both of these pathways, the first type of the caspases that are activated, they are known as the initiator caspases, and these initiator caspases then are going for the activation of the another class of the caspases which are called as the executional caspases. And actually these are the executional caspases which kill the cell by degrading the protein indiscriminately, but these executional caspases, they are actually activated by the initiator caspases. In the extrinsic pathway, the apoptosis that is triggered because of the external stimuli. For example, there is viral infection in the cell. So when there is a viral infection in the cell, that means that the virus is coming from the exterior of the environment and it is infecting the cell, therefore is giving a signal for the apoptosis of those particular cells. So when there is viral infection, specific ligand that binds to these virally infected cells, to the receptors that are present on the surface of these cells, and these receptors are typically members of the a large family of the receptors which is known as the tumor necrosis factor receptor gene family. For example, one of the important receptor in this family is known as the tumor necrosis factor receptor 1, or another important one in this particular family is known as the FAS receptor. And when the ligand that binds to these receptors, that actually leads to the activation of the caspases. Now let us discuss the extrinsic pathway in a little bit of detail, how these death receptors work and how the ligand binding is actually responsible for the activation of these death receptors on the cell surface. On the surface of all of the cells, you have got these FAS receptors or these death receptors. If you talk about this FAS receptor, it have got an extracellular domain which is responsible for the binding of the ligand, and you have got the intracellular domain which are actually responsible for the activation of the caspases indirectly. Initially, when the extracellular domain of this FAS receptor is not bound by a ligand, that means that the interior domain, which is known as the FAS associated death domain, for short you can call it as the FADD, or simply you can call that as the death domain. So when there is no receptor bind to the extracellular domain of the FAS receptor, this FADD domain, it remains inactive. That means that the downstream pathways, they are off. When there is when a ligand that binds to this particular FAS receptor, that is actually going for conformational changes in this intracellular domain, and when there are conformational changes in the intracellular domain, that actually leads to the activation of the downstream proteins or the downstream receptors. One of the important things to keep in mind is that when you talk about the cellular signaling and you talk about the ligands and the receptors, usually these ligands, they are freely available in the extracellular domain. They are freely moving in the extracellular in the extracellular environment. But these FAS receptors, the ligand for these FAS receptors are known as the FAS ligand. And these FAS ligand are actually bind to a particular cell, for example, the lymphocytes. For example, when you talk about a T lymphocyte. So if this is a FAS ligand, this means that this FAS ligand is actually attached to a T cell, and the T cell is actually attaching to this particular FAS receptor. Initially, when you talk generally about the ligands, these ligands are actually freely in the extracellular matrix. That means that only the ligand is binding to the receptor. But in this particular case, the ligand that is attached to a T cell, and when the ligand bind to this receptor, that means that whole of the cell that is actually binding to this particular receptor. So when the FAS ligand that binds to the extracellular domain of the FAS receptor, the intracellular domain of the FAS receptor, that is going to get activated. There is another important protein in the cytoplasm of the cell which is called is the phase-associated death domain adaptor protein, and for short you can call that is the FADD adapter protein. If you talk about the structure of this particular FADD adapter protein, just like the phase receptor, it also have a phase-associated death domain. That means that these two, they are very similar to each other. You have got FADD domain in the phase receptor. You also have FADD domain in the FADD adapter protein. Another important domain that is present in the FADD adapter protein is known as the death effector domain, and for short you can call that as the DED. So when the ligand that binds to the receptor causing conformational changes in the FADD domain of the phase receptor, that actually leads to the binding of this FADD domain to this FADD domain. As you can see this in this particular diagram, that the FADD domain of the phase receptor that is going to interact with the FADD or the dead domain of the FADD adapter protein. So initially they are not interacting with each other, but when the ligand binds to this particular receptor, these two FADD domain, they are going to come attached to each other. They are going to attach to each other, and the death domain, the DED domain that you can see over here, now that is free, that is still free in the cytoplasm of the cell. Another important protein that is present in the cytoplasm, that is known as procaspase 8 or the procaspase 10. So this diagram is actually representing a procaspase 8, or it is also representing a procaspase 10. So it can be 8 or it can be 10. If you talk about the structure of this procaspase, it have got 4 different domains. One of the important domain that is known as the DED domain or the death effector domain, that is also present in the FADD adapter protein. So this domain and this domain, they are actually very similar to each other. Another important domain that is present in this procaspase, that is known as the pro domain. You have got a large subunit over here and you have got a small subunit over here. Now this diagram, this one, the procaspase 8 or the procaspase 10, that is actually the inactive form of this particular caspase. Therefore it is known as the procaspase. Another important thing to keep in mind is that this procaspase 8 or this procaspase 10, it is also known as the initiator caspases. We will come to that in a while, that what are the initiator caspases and what are the executional caspases. But for this particular moment, keep in mind that we are talking about an initiator caspase over here. So when the phase receptor that interacts with the FADD adapter protein utilizing its FADD domain, that is actually activating this DED domain of the FADD adapter protein to interact with this procaspase 1. You can see that in this particular diagram, that the DED domain of the FADD adapter protein that is going to interact with the DED of the procaspase. So initially the DED domain of the FADD adapter protein that is not in interaction with the DED domain of the procaspase, but when the FADD domain of the phase receptor that interacts with the FADD domain of this adapter protein, it is actually going for the interaction of this DED domain of the FADD adapter protein to the DED domain of the procaspase. So when the DED of the FADD adapter protein that interacts with the DED domain of the you can say this procaspase, you are actually having this very large complex, and this whole complex, it is known as the death inducing signaling complex, and for short you can call that as the DISC. This means that the phase receptor binded to the ligand, the FADD domain that is interacting with the adapter protein, the DED domain of the adapter protein is interacting with the procaspase. So this whole complex, all of them, the receptor, the adapter protein and the procaspase, this whole complex is known as the death-inducing signaling complex. Now what this death-inducing signaling complex do is that it is going to cleave this procaspase at two sites. It is going to make one cut over here at this particular site, that is in the intersection of the prodomain and the large subunit, and it is going to cut the prodomain at this particular site, that is the intersection between the large subunit and the small subunit. So if you are going to cut the procaspase at this particular point, and if you are going to cut this at this particular point, you are actually separating the large subunit and the small subunit from each other. You can see that over here, that the large subunit and the small subunit, now they are separated from each other. Initially they are in the procaspase form, they are attached to each other, but this death-inducing signaling complex is actually going to cut the large subunit and the small subunit from each other, thereby making the large subunit and the small subunit separated from each other. Now, to make an active initiator caspase, as I've told you, this particular form of the caspase is the inactive form, or the pro form of the initiator caspases. But when you are interested in making the active initiator caspases during the process of the apoptosis, you need to make two large subunits and you need two small subunits. So if you look at this one, this is just a very simple one. You are actually focusing on a single procaspase, but in reality, a lot of the Fas receptors that are present on the surface of the cell, a lot of them, they are making a very large. You can say they are making a very large number of the. They are cutting a very large number of the procaspases, generating a large amount of the large in the small subunit. So two large and two small subunits, they combine with each other, and when they combine with each other, they are actually going to make an active initiator caspases. And these active initiator caspases, what they do is that they are actually going for the activation of the executional caspases. If you talk about the executional caspases, the structure of the executional caspases and the executional and the initiator caspases, they are very much similar to each other, and the only difference is that there is no DED domain in the executional caspases, and they only have the prodomain, the large subunit, and the small subunit. What this means is that the if you compare the executioner and the initiator caspases to each other, the only difference is this DED domain. This DED domain that is present in the initiator caspases, and this is absent in the executioner caspases. So once the initiator caspases, they are activated, what they are going to do is that they are going to cut the executioner caspases at two sites. They are going to cut at the intersection of the prodomain and the large subunit, thereby making one cut over here, and they are also cutting at the intersection of the large and the small subunit. In this particular process, again, just like the initiator caspases and the executioner caspases, you will be having a large subunit and you will be having the small subunit. And again, just like the initiator caspases, two large subunits and two small subunit, they are going to combine with each other to give you an active executioner caspases. Now, what are these caspases then? As you can see that we have activated the initiator caspases with the help of the binding of the Fas ligand to the receptor, activating the DED domain, and those the death-inducing signaling complex is actually making the active initiator caspases, and the initiator caspases are then going for the activation of the executioner caspases. So what are these caspases? As I've told you, these caspases, they are a family of the protease enzymes, and they play a very essential role in the process of the apoptosis. Now, why they are called as the caspases then? These caspases, they are known as the caspases because if you look at their active site, in their active site, they have got a specific cysteine amino acid, and this cysteine amino acid is very important for their enzymatic activity. So if you remove this cysteine from their active site, these enzymes, they are not going to work then. So this means that this cysteine is required for their enzymatic activity. Therefore they are called is the cysteine dependent. Another important thing is that they are aspartate directed, and they have got cysteine in the active site, and they cleaves a target protein only after an aspartic acid residues. So this caspase name, if you look at this particular name, this C is actually the coding for the cysteine dependent, this ASP is for the aspartate directed, and this ASES is actually referring to the Proteus. So they are called is the caspases because they have got cysteine in their active site, and they cleave a target protein only after an aspartic acid residues. There are about 11 or 12 confirmed caspases in the human and 10 in mice, and these particular caspases are carrying out a variety of the cellular functions. These caspases, as I've told you, they are broadly classified into two classes. One is known as the initiator caspases, and the example of the initiator caspases, that is caspase 2, 9 and 10. These are the examples of the initiator caspases. When you talk about the executioner caspases, the important examples are the caspases 3, 6 and 7. And once the initiator caspases they are activated, they are actually going for the activation of the executioner caspases, and these executioner caspases are going to degrade over 600 cellular component in order to induce the morphological changes in the apoptosis that we discussed in the start of the presentation about the blabbing, about the global messenger RNA decay and all those sort of stuff. If you talk about the important target of the executioner caspases, one of the important target of the executioner caspases is an inhibitor of the DNase. You all know that the DNases, they are actually responsible for the fragmentation of the nuclear DNA. But in normal cases, when the apoptosis has not been started before the apoptosis, this DNase is actually in interaction with an inhibitor, and this inhibitor is inhibiting the activity of the DNase. Therefore the nuclear DNA is not degraded. But during the process of the apoptosis, these executioner caspases that degrade that inhibitor of the DNase, and when the DNase is released from its inhibitor, it is going for the fragmentation of the nuclear DNA. Another important target of these executioner caspases are the nuclear lamins. And these nuclear lamins, they are very important protein when you talk about the structure or the morphology or the stability of the nuclear membranes. So these nuclear lamins, they are degraded by these executioner caspases, and when these nuclear lamins they are degraded, that actually leads to the fragmentation of the nucleus. Another important target of these executioner caspases, they are the cytoskeleton proteins. The important cytoskeleton proteins, they are the actin, the myosin, the tubulin, etc. So when these executioner caspases they degrade these cytoskeleton protein, there is a disruption of the cytoskeleton, there is membrane blabbing, and hence the fragmentation of the cell. Another important target of these executioner caspases, they are the Golgi matrix proteins, and when these Golgi matrix proteins they are degraded by the executioner caspases, that actually leads to the fragmentation of the Golgi apparatus. So this is how the executioner caspases they are going for the degradation of the cell during the process of the apoptosis.